Hello, I'm Mark Knepper, Chief of the Laboratory of Kidney and Electrolyte Metabolism at the National Heart, Lung, and Blood Institute in Bethesda, Maryland. Today it's my pleasure to interview Maurice Burg about his nearly 50-year career rather, in science. Dr. Burg is principal investigator in the Laboratory of Kidney and Electrolyte Metabolism at the National Institutes of Health in Bethesda. Throughout his career, Dr. Burg has been a leader in the field of renal physiology. He's known for the development of critical technology that has opened new paths to discovery of many of the physiological principles that comprise our modern understanding of renal function and osmotic regulation. Not only will we hear today about the invention of the isolated perfused tubule technique, for which he's best known, but we'll also hear about a host of other innovations basic to the study of kidney physiology. Now, this interview is being conducted on May 10th, 2005 at the National Institutes of Health in Bethesda, Maryland. The producer is Alice Hardy. Copies of the interview will be distributed to the American Physiological Society for its eminent physiologist archives and to the International Society of Nephrology for its legacy archive. A copy will also be deposited in the office of the NIH History here in Bethesda. Now, sitting on my right is Dr. Burg, known to his friends as Mo. Let's start at the beginning. Did you have any early interest in science?
Sadly, no. Those were before the days of science fairs and that kind of effort to attract people to science. I did enjoy my science courses in high school, but no more so, for example, than English courses. And in college, my major was psychology, which in retrospect was hardly scientific, at least in those days.
So why did you decide to go to medical school?
The choices seemed to be between a career in medicine and a career in law. My father wanted me to go into law. Maybe that's why I ended up in medicine. That's not clear right now. Anyhow, I kept my options open in college, and I found that I was enjoying my pre-med courses. The die was cast finally when, after 3 years in college, I applied to Harvard Medical School, and they accepted me, and so I ended up in medical school at that point.
So how did you become interested in kidney physiology then?
Well, more or less by accident. Being a psychology major seemed natural to go into psychiatry after medical school, but I had classes in medical school, in psychiatry, and clinics, and it all seemed pretty futile at that point. And so I turned my interest to more traditional kinds of medicine. After medical school, I was an intern at the Beth Israel Hospital in Boston, and in those days we had a draft for doctors. The draft was called the Berry Plan, and you had an interesting choice at that time. The choices were you could either volunteer to go into the Army Medical Corps, in which case you went in as an officer, or you could not volunteer, in which case you would be drafted as a private. Most physicians volunteered, as I did. And so while an intern at the Beth Israel Hospital, I had my orders to report to Fort Sam Houston for basic training in the Army and the Army Medical Corps. However, about 2/3 of the way through the internship, I got a telegram from the Army, and the telegram said that they had over-enlisted in the Berry Plan that year, and if I could find a medical residency, they would permit me to take that medical residency before serving in the Army. That seemed like a good deal, so I went scurrying around Boston looking for a medical residency at that late time. And where I ended up was at the Boston VA Hospital, which you can see some of the personnel here in this first slide. When I first went there for an interview, the interviewer was Dan Holtzman, who was the head of cardiology. Now, while the Beth Israel had a very strong program in cardiology, and that was really what interested me at that point, so I was pleased to go into a residency in cardiology. However, when I arrived at the VA hospital to begin my cardiac residency, I discovered that I was a resident in kidney and endocrine. And so I immediately began seeing consults in kidney disease and endocrine disease. Now, the chief of medicine in the hospital was Maurice Strauss, who's a very charismatic person, great teacher. And his interest was kidney physiology. And so he had a kidney physiology program going in the hospital at that time. The people who were mainly responsible for that were two physicians, Saul Papper and Jack Rosenbaum. The kidney physiology consisted of clearance experiments in veterans. And so I was soon involved in infusing veterans with various solutions, with inulin and under various conditions. Saul went on to become chief of medicine at in Albuquerque, New Mexico. Jack sadly developed colon cancer while I was at the VA hospital and died. He was a very promising kidney physiologist at that time. It was very sad for all of us. Now, as a result of my kidney physiology at the VA, there were two papers. This is the second one, which is the factors influencing the diuretic response to ingested water. And by that time, there were already published works by Wirtz, which indicated that fluid in the early distal tubule should be diluted. And so the idea that the free water clearance, which we were studying, might be regulated by sodium delivery to those regions of the kidney were already understood, and that was more or less the conclusions that we came to in this paper. It wasn't until many, many years later that I had the opportunity to actually look at the mechanisms involved and describe the transport mechanisms in the kidney that were responsible.
So that was in your hometown in Boston. How did you happen to come to the NIH?
Halfway through my residency at the VA hospital, again, there was a serendipitous occurrence. Maurice Strauss came to me and said that he had contacts at the National Institutes of Health, and how would I like to become a public health officer and do research at the National Institutes of Health instead of being a medical officer in the Army and treating the soldiers. That seemed like a very good deal to me. So I volunteered to go to NIH, which fulfilled my requirement under the Berry plan. Okay,
then why have you stayed at NIH so long after your postdoctoral fellowship there?
I first came to NIH in 1957, and so I was there for 2 years initially as a fellow. Then I went back for a third year of residency to the VA. I think by that point I had already decided that I liked research very much, so that during my residency at the VA, I was trying to decide whether I wanted to continue there or whether I wanted to return to NIH, since Jack Orloff, who was my mentor at NIH, had already mentioned to me that if I wanted to come back, I could. Well, there was no comparison between the research opportunities at the VA and the research opportunities at the NIH, so I very quickly elected to go back to the NIH. And the research was marvelous, NIH was marvelous, and I've never found reason to leave since then.
So this was the kidney laboratory of kidney and electrolyte metabolism that you first went to. What was it like when you arrived in the laboratory in 1957?
I think that's best described in terms of the people who were there, which is on the next slide. In those days, there weren't many pictures being taken in the laboratory. Nowadays, we take a picture every year with all of the lab members. So this is the closest I can come to picturing the people who were there. The occasion was a dinner given for Bob Berliner many years later in Atlantic City. But many of the principal people I want to talk about are represented in this picture. Central to it all is Berliner and Jack Orloff. And those were the principal people in the laboratory, and they set the tone in the laboratory. They were both brilliant critics, very bright people, and it was an education just to listen to them and to discuss the various aspects of kidney physiology. Even more impressive, they were very widely respected in the kidney community, and even, I think, feared a little bit because they were such powerful critics. A memory goes back to Atlantic City. In those days, the principal meetings for research scientists in renal physiology were in Atlantic City. The first meeting being the clinical societies at the Haddon Hotel, and the second meeting being the Physiology Society at the Convention Center. And of course there were sessions at the meetings, but a lot of what was most important went on the boardwalk outside of the meetings. And I can recall Berliner holding court on the boardwalk and having very eminent investigators come up to him to tell him about their ideas and their work and to get his view on it. And his view was very important to them in what they should make of it themselves. In the meetings, you would have a 10-minute talk and then 5 minutes of discussion in which people would ask questions or make criticisms. I think there was a gripping of fear on the young fellows' faces when Berliner would stand up to ask his question or make his criticism of their work. The laboratory of kidney electrolyte metabolism had begun in 1948. I didn't arrive there till 1957. The first director of the Heart Institute was James Shannon, who was a famous kidney physiologist. When he staffed the Heart Institute, the first laboratory that he staffed, or one of the first laboratories, was the Laboratory of Kidney and Electrolyte Metabolism, which remains the longest established laboratory in the Heart Institute. Why a kidney laboratory in the Heart Institute? Well, Shannon realized that the most important cardiac diseases involve the kidney: congestive failure, hypertension, salt retention, and so on. And he recruited Berliner, who was a brilliant young kidney physiologist at that point, to head the laboratory. So Berliner was the first lab chief, but he was only lab chief for a couple of years. He went on to become director of the Heart Institute intramural, and Jack Orloff took over as the lab chief at that point. They had very different personalities. Berliner was the quiet sort. You had to sort of press him to get his opinions. Jack was very outspoken, very flamboyant. If you wanted to have a party, you would have Jack there as the life of the party. Jack also had a very abrasive wit. he was prone to make lacerating comments to people in public. And this was done in a way that was in a joking way, so that you couldn't really respond to it without being a poor sport. And it was generally quite funny to everyone, except possibly the person who was the brunt of the joke. At that time, the principal event, which is memorable in the laboratory, was our lunches. And so we had lunch every day as a laboratory group. And the lunches, there was a paper presented by one of the fellows. And Orloff and Berliner, who were the professors, would comment on the papers. And this was quite educational. They were very knowledgeable and very good critics, and so you could learn a lot from it. In retrospect, one of the aspects of it, which I wasn't really aware of at the time, but I'm aware of now with our new sensibilities, was that the lunches were an entirely male affair at the time that I was there. Well, all of the fellows were male, so that made sense. The women in the laboratory were the technicians, Nordica Green and Agnes Preston. And I'm not sure whether they weren't included because they were technicians or because they were women. At that time, you may recall that when you published a paper, you would have a list of the real authors on the paper, and then it would say, with the technical assistance of, and it would list the technicians, who often were the ones that did the actual work. We no longer do that, and I never did it. I think the first woman to ever sit in one of these conferences was a summer student of mine, Evelyn Grohlman. And there was a bit of resistance to her sitting in, and I'm not sure whether it was because she was a student or whether she was a woman. There was a bit of the old guy's attitude at these conferences, and I think that Jack feared that he would have to be more discreet with his language if there were a woman present than if there were not. At that time in the laboratory, transport physiology was in its infancy. The only transporter that was really known and was of interest to us was the sodium-potassium ATPase that had been recently discovered by Sco, who, you remember, recently got the Nobel Prize for that discovery. And I think the best transport work in the laboratory was work on red blood cell transport, which was being carried out by Dan Tosteson when I first arrived there, and Joe Hoffman. And then when Dan left to go on to his very eminent career in medicine and medical administration, Joe Hoffman remained on for many years, and I learned a lot from Joe. The kidney studies that were going on in the laboratory were largely clearance experiments, and those clearance experiments were mostly carried out in dogs. I did one dog clearance experiment in my career, and then, for whatever reason, decided not to do any more, and ended up with other preparations. I was Jack Orloff's fellow. The other fellow at that time was Floyd Rector, who's also in this picture. Floyd left after my first year in the laboratory to go to Dallas, where he had had a very eminent career. The desk I sat at when I first arrived had been occupied by Mac Walser, who went on to become the professor of pharmacology at Johns Hopkins University. I became quite close to Floyd during that first year. We shared a lot of interests. We spent a lot of time talking. I can remember the two of us trying to sit down and figure how a kidney tubule could have regulated absorption of sodium based only on the sodium-potassium ATPase. And we didn't come up with any answer to that because, of course, much more is required, which was the later work that both of us did. Of course, they'll spend a lot of time there and a lot of memories. One which is particularly striking has to do with the urinary concentrating mechanism. This was an interest of Berliner's, and it was at the time when the countercurrent mechanism had been described by Haggerty and Kuhn and Wirtz. And so the puzzle at that point was exactly how the transport processes in the tubule segments in the medulla worked in the countercurrent system. Berliner's idea was that it operated strictly by transport of salt out of the tubule, leaving water behind, which nowadays we would recognize as the avian model of medullary countercurrent multiplication. And he and Norm Levinsky had some clearance experiments that seemed to support that idea, which they submitted as an abstract to the meetings in Atlantic City. However, after they had submitted the abstract, Karl Gottschalk arrived in the laboratory, and he had already at that point made his seminal discoveries regarding the concentrating mechanism and knew that the fluid within the loop of Henle, rather than being dilute according to the Berliner theory, was concentrated, as we now know. This was not very good news for Norm Levinsky, since he was then faced with presenting an abstract at Atlantic City in which the conclusion was incorrect.
So as a bright young scientist coming to the lab in 1957, what was your initial project?
My first project was one that was assigned to me by Jack Orloff. And the question was, is the sodium-potassium ATPase important for transport in the kidney? And the way of answering that was to use a specific inhibitor, which in that case was strephanthidin. And the preparation that we chose to study was the renal portal circulation of the chicken. This is a sketch I made at the time in order to try to understand it, which I won't go over in detail, but the essence of it is that if you infuse a solution into a vein in one leg of a bird or a chicken, in this case, it will go through a renal portal circulation and result in much higher concentrations of what you infuse around the kidney tubules and the kidney on that side than on the other. So you can have a experimental on one side and control on the other side by collecting the fluid separately from the two ureters. Now, I didn't know at the time, but this project had first been offered to Floyd Rector, and Rector refused to do it. I was neither knowledgeable enough nor assertive enough to refuse to do it. There were two results from the experiment. The first result was what we published, that the sodium-potassium ATPase is very important for salt absorption in the kidney. And this was a novel discovery at that point. The second discovery was that the chicken cloaca, where the ureters come out, is a very filthy place, and you don't want to be working in there if you can avoid it.
Okay. What happened next?
Well, Jack wanted me to study the sodium-potassium ATPase. I was not a biochemist. I had no biochemical training. And frankly, I didn't understand at all what it was biochemically. It was confused in my mind with the ATPase and the mitochondria. I tried reading about it. Jack was not much help to me. And I finally decided that that was not the thing that I was going to study. So I decided instead to study renal transport in vitro, that is, outside of the animal, having already tried a clearance experiment on dogs and not having enjoyed the experience very much. Now, there were good reasons at that time to look to in vitro preparations. One was that the most impressive epithelial transport studies at that time were the studies in frog skin coming out of the Ussing laboratory. And it looked as if that could be carried over in some respects to the kidney, because there were already descriptions of experiments on the iron contents of kidney slices, which are carried out by Gilbert Mudge at Johns Hopkins. And there were also experiments showing that kidney tubules could survive in vitro from a Chambers laboratory. So I adopted Mudge's kidney slice technique and did some studies of the effect of warbane on the electrolyte content and other aspects of the kidney slices. And in retrospect, none of them were terribly important results.
Mm-hmm. And so why did you abandon the kidney slice preparation?
I realized that that when you were studying particularly the kinetics of the electrolytes in kidney slices, that there were almost impossible problems, because the kidney slices were relatively thick preparations, many tubules thick, so that there was a diffusion delay for anything that you put into the solutions to get to the surfaces of the tubules. The slices, particularly from the cortex, are thick enough so that the oxygenation of the tubules in the center is not very good. And also, you have all sorts of different tubules there, which means you have different kinds of cells. So I first set out to try to solve the problem of the thickness of the kidney slices, which led to the next slide, which is the preparation of kidney tubules. This had never been done up to that point, and I researched a way of doing it by looking at papers in cell culture, in which they used enzymes, including collagenase, to disaggregate tissues to start the cells in culture. So I tested collagenase, trypsin, various other enzymes to see if there are any that would dissociate the kidney into tubules that would be surviving, and ultimately devised a method using collagenase to make tubule preparations such as this shown here from the cortex of rabbit kidneys. Now, the rabbit was chosen mainly because that was the species I've been working with in the kidney slices, and it's a good species to work with if you want to study sodium-potassium ATPase, because the ATPase in the rabbits is quite sensitive to cardiac glycosides, whereas the ATPase in rats' kidneys, for example, is not. Did a lot of studies with the suspension over a couple of years. The tubule suspensions have been widely used since then. It seems that each person who used it after that, starting with my method, invented it afresh and forgot whether they had gotten the method from. But I think that's the way science goes.
Well, back in those days, the cutting-edge technique was considered to be micropuncture. Did you consider doing micropuncture?
Oh, yeah. Floyd Rector and I discussed that a lot, and I wanted to do micropuncture, but I couldn't. I couldn't do it because Berliner wanted to do micropuncture, and of course he was the professor and decided what went on in the laboratory. And he had assigned Tom Kennedy, who's was then in the laboratory, to set up micropuncture for him. And when I left the laboratory after my first 2 years of a fellowship there, Tom Kennedy was supposed to be setting up to do the micropuncture. By the time I returned a year later, Kennedy was no longer there. He had gone into administration, and there was no micropuncture. Rector, who had gone on to Dallas, had started the micropuncture there, and the person who was doing it there was James Clapp. And after Jim finished his time in Dallas, he came to NIH, and he is the one that set up the micropuncture at NIH in Berliner's laboratory. Berliner chose to do micropuncture in dogs, I guess in part because that's where the clearance experience was and where the knowledge was. I don't think that that was a particularly good choice. I don't think it's a very practical preparation for doing micropuncture. So I couldn't do micropuncture, and instead, by the time I left NIH after my first 2 years of fellowship, I had decided that the way to study the kidney tubules was to take them out of the kidney and perfuse them in vitro, which of course was a rather audacious idea at that time. It seemed to me that it was possible. The examples I had, which brought it to mind, were first the giant squid axon that Hodgkin and Huxley had studied and had famously published by that time. Now, a squid axon is a millimeter in diameter, whereas a kidney tubule is only 1/20th of that diameter, so there's a big size difference. However, you looked at the pictures of the axial electrodes in the squid axons and all of the marvelous transport studies, that had to be very attractive. Also, I knew that Chambers had made preparations of tubules from fish and amphibia, and that those tubules survived in vitro to at least transport dyes, so they were still alive. So why not try to dissociate single tubules and to perfuse them.
So how did you learn to dissect these tubules?
Well, I tried to figure out how to do that myself. I already had the example of the collagenase, and so it was natural to start with the kidney preparation in which the tissue had been treated with collagenase, and it's relatively easy to dissect tubules out. But I didn't know that at the time, and I started with a very cumbersome system, which I was using micromanipulators to try to do the dissection. And it wasn't until we had a visitor to the laboratory, who was Ivar Sperber, that I really found out how to dissect. Now, Ivar Sperber was a famous renal anatomist. He, you may recall, is the one who had discovered the portal circulation in the bird, and which was the first preparation I used. And he had also dissected kidneys and shown all of the tubule segments, which you can see here. Of course, these dissections were on dead tissue that was macerated with strong hydrochloric acid, and I was trying to dissect more delicate tubules from living specimens. Well, Sperber came to visit the laboratory to give a lecture, and while he was there, he showed me how to dissect tubules, which is with forceps and needles rather than with micromanipulators, and it turned out to be much easier to do it that way. While he was there, Sperber gave a lecture which was very memorable. While he was lecturing, a messenger kept coming into the room and speaking to Berliner, who would shake his head. At the end of the lecture, Berliner announced that President Kennedy had been shot, and we all rushed out after that to look at the television and see what had happened to the president. I think those of us who were old enough at the time all remember where we were when we got that news.
November 22nd, 1963.
You remember it? Well, so I, at that point, had learned how to dissect the tubules. What happened after that was that I had my first fellow arrive, who was Maurice Abramov. Maurice is Belgian, and he arrived at the time I was working with the suspensions of kidney tubules from the cortex, and was doing experiments on the kinetics of sodium and potassium transport in the tubule preparation using radioisotopes of sodium and of potassium. The one aspect of that was my first real introduction to kinetics, and compartmental analysis was evidently required. At the time, the first computers, big mainframe computers, were available, and we had at NIH a mathematician who knew how to program that for kinetic analysis, Maurice Burg. And so I went to Maurice, and he set up the kinetic analysis system with the computers, punched the cards. I think you're old enough to remember the punch cards in the mainframe computers, and solved our kinetic problems. And Maurice and I used the suspension to unravel some of the aspects that were mysterious in the kinetics in the tissue slice, but still we weren't where we wanted to be, because in the tubule suspension, the main surface that's available to the fluid is on the outside of the tubule, and what we're really interested in was transepithelial transport that requires access to the lumen of the tubule and requires perfusion of the tubules.
So how did you figure out how to perfuse the tubule? Sounds impossible.
Joe Hoffman was a big influence in that. He's shown here. This is much later picture taken at the time of our laboratory reunion. Joe was in the lab for a long time and eventually left to become a professor of physiology at Yale. Well, I first needed pipettes to get into the tubule lumen. Joe had a microforge. He let me use his microforge to make perfusion pipettes. And it's not easy to poke a perfusion pipette into the end of a piece of tubule that is floating around. And I tried holding the tubule in various ways with little suction pipettes around the outside to open it, and none of that worked. And I finally realized that I needed suction all around the tubule, which meant I needed a concentric pipettes. The outer pipette to apply the suction, the inner pipette to have the suction pull the tubule over that and to perfuse the tubule. Well, at that time, the electrical potential in the red blood cell was had not yet been measured, and Joe wanted to measure that along with Walter Freigang, who was a neurophysiologist at NIH, and so they made a special apparatus which held pipettes concentrically, in which the inner pipette was intended to hold the red blood cell, and the outer pipette was intended to flow sucrose over the cell so they could use the sucrose gap method to measure potential. It didn't work. And when it didn't work, Joe willed that apparatus to me, which was the holder for concentric pipettes that I first used to perfuse tubules. So using that, I successfully perfused my first proximal straight tubule from a collagenase suspension, as shown next here, and the result was rather dramatic. The tubule exploded. The problem, of course, was that the collagenase had removed the basement membrane, which is the main support for the tubule, and so it could not withstand the pressure inside, and it was obviously necessary to dissect the tubules without collagenase. That turned out to be not not that difficult with the not that difficult from the rabbit kidneys, and used rabbit kidneys for the initial perfusion studies and for a long time thereafter. In retrospect, it was clear that the reason that the rabbit kidneys were so easy to perfuse was that, unknown to me at the time, NIH had a very special colony of rabbits from which I was getting the rabbits that I used, and that colony of rabbits was pathogen-free. Ordinary rabbits from a farm are not pathogen-free. They have kidney infections with various parasites, and that increases the fibrous content of the kidneys, and the tubules are much harder to perfuse. So at this point, I had tubules that I could dissect and a way of perfusing them.
And then you needed to collect the fluid. How did you achieve that?
There I was lucky. My second fellow was Jared Grantham, who's shown here many years later. Jared is a very capable scientist and investigator. He went on after his fellowship with me to return to Kansas City, where he originated, becoming eventually the chief of nephrology, and has remarkable accomplishments with respect to polycystic kidney disease, in which he started a foundation. He has a lot of studies himself, and I think if anyone is responsible for progress in that field, it's Jared. And underneath his portrait here, you can see that there is a pipette at the other end of the tubule. The perfusion pipettes are at the right-hand side, showing the inner perfusion pipette and the suction pipette. And at the other end, he simply had one pipette that he sucked the tubule into, had oil in the pipette, and introduced a collection pipette to collect the fluid as it accumulated.
Mm-hmm. And this Hoffman-Freigang concentric pipette apparatus, did you continue to use that?
No, that wasn't very practical. And the thing that it lacked was, although it held pipettes concentrically, adjusting them one within the other was very tedious. And what was obviously needed was a micrometer arrangement so that the inner pipette could be advanced relative to the outer pipette. Gerhard Giebisch knew what I was doing at that time, and he recommended that I go to Johns Hopkins and visit a laboratory there, which was run by Philip Davies, because Philip Davies had such an apparatus that he was using for other purposes. So Maurice, Abramov, Jared Grantham, and I went to visit Philip Davies, and sure enough, he had an apparatus that would do exactly what we wanted. However, it was a very delicate apparatus that he essentially assembled each time he used it, and it didn't seem to me to be very practical for continued use in perfusing tubules. So I wanted to have a more practical arrangement, and that in order to do that, I had a choice. I could have gone to the engineers who were at NIH and had them design it, but I had no faith that the engineers would design something that was both practical and durable. So I went directly to the mechanics at the NIH shops and visited with Ken Bolin. Now, Ken was the head of the optical section, so-called, of the shop, which is where they had a few of the mechanics or the machinists who were most highly skilled. And Ken himself was a watchmaker, so this is the sort of thing that would be very good. So I sat down with Ken and with one of the very good machinists in his shop, who was Jim White. And they designed the apparatus that you can see here, which was our first practical apparatus for perfusing tubules. Jim retired from the shop a while after that and devoted himself entirely to constructing perfusion apparati for all of the laboratories around the world, including our own, that wanted to have tubule perfusion studies. Now, Jared used this apparatus, which you see to the right here in one of his early experiments, to hold the perfusion pipettes. And at the other end, you see the only existing picture of the original Hoffman and Freigang concentric pipette holder, which in this case Jared was using simply to hold the perfusion pipette to collect the fluid.
And then what improvements were made subsequently to the apparatus?
Well, once we're able to dissect and perfuse kidney tubules, there were lots of studies we wanted to do. One thing was to measure fluid absorption. Initially, we used I-131 albumin as a marker. Later, we and others used inulin, radioactive, and such things. You developed, I think, the best method, which is to use fluorescently labeled markers and get away from the radioactivity altogether. Once we had those measurements, we needed to do the mathematics to calculate the fluid absorption rate. I'm not much of a mathematician, so I went to our very good mathematician at NIH, who was Clifford Patlack. A visit to Patlack's office was an interesting experience. In his office, every surface was covered with piles of manuscripts and papers, no free surfaces at all. The entire office is surrounded by blackboards, and the blackboards were entirely filled with equations. You were allowed to erase just enough of the blackboard to put down your problem. Then Clifford would stand up and go to the blackboard and would derive the equations that were needed for your project. You'd write them down, you'd take them back, and proceed with them. Clifford was very remarkable. He was a mathematician who was also a physiologist. He understood the mathematical requirements of physiologists, and you could talk to him directly since he was as interested in the physiology and the applications as in the mathematics itself, which is not true of many mathematicians. Clifford went on to become a professor at Stony Brook in New York. We needed methods to measure the ions, organic compounds, and others in the collected tubule fluid. There we were very fortunate to have within the Heart Institute the Laboratory of Technical Development, which was headed by Robert Bowman, a remarkable person, very colorful character. He was very creative and inventive, and he had in his laboratory Jerry Vurek, who was the principal person who helped us. Together they developed the radio frequency photometer that we use for measuring sodium and potassium. Vurek developed the method for measuring total CO2 by calorimetry. He developed very micro fluorimeters and colorimeters, and you became the guru of the microfluorimeter and the micro colorimeter, adapting them to all sorts of assays, which you eventually used. Also, being able to take the kidney apart and dissect the different tubule segments opened up all of the tubule segments to study. Micropuncture was marvelous for studying the tubules on the surface of the kidney, but there are very important things happening in tubules within the kidney. And so we developed methods for studying all of the segments eventually in the kidney. Also started with rabbits, which were easy to dissect, not perhaps the most practical animal for studying, particularly since so much of the physiology study had been done on rats previously. And I tried dissecting rats and decided it was too hard and gave it up. You said you wanted to dissect rats. I told you it was impossible. but being a good investigator, you didn't listen to me. And you realized that getting pathogen-free rats would be very important, just as it was with the rabbits, and you succeeded brilliantly with that.
So a lot of your early studies were on ion transport, which required electrical measurements. How did you achieve those types of measurements?
Well, one of the electrophysiologists and transport physiologists was Jack Dainty. And when I presented my work at a meeting that he was at, he criticized it. He said, you know, you can't interpret these results without knowing the electrical potential. I realized at that point that that could be fairly easy to measure, since I could use the perfusion pipette as a bridge into the tubule lumen. And so I did it that way. Then I ran into another problem, and that is liquid junction potentials, which I, of course, had never heard of. But and another one of my presentations and meetings, I ran into Jared Diamond, who was a brilliant young transport physiologist. Jared, of course, is now the most famous of us all, having recently won the Pulitzer Prize for his work on guns, germs, and steel. And while he was a very good physiologist, he was also even a better ornithologist and evolutionary biologist. And his interest was history, so he's now in the geography department at UCLA. Anyhow, Jared was a very good electrophysiologist. He explained liquid junction potentials to me, and he explained how you measure them and how you adjust for them. And so we made those adjustments and did our first measurements of tubule potential. Measurements are not shown here, but the fellow is shown here. This is Leon Isaacson, who came to me from South Africa as one of the early fellows. This is many, many years later, after his retirement from being a professor in South Africa. I didn't realize it at the time, but Leon, at the time he came to be my fellow, was already much older than I was. So I was instructing older people. And Leon used the electrical system to measure the voltage across collecting ducts, cortical collecting ducts, which we were studying then. Further advance came when Sandy Hellman arrived, and Sandy was an electrical engineer, which is really just what we needed. I know you're overly fond of engineers, but this was one that paid off for me. When he left the laboratory, went on to become a professor of physiology at in Indiana, and very good, and continued his electrophysiological studies there. His job as my fellow was to measure the resistance of the kidney tubules. In order to measure resistance of tubules, we used cable analysis, of which I was totally ignorant, but Sandy, being an electrical engineer, was very familiar with it. The original concept having come, I think, from Lord Kelvin with relation to laying the Atlantic telephone cables, and applied extensively in neurophysiological research with the squid giant axons and such. Anyhow, Sandy knew the cable equations, he knew how to do the measurements, and so he was able to measure the electrical potential both in mainly in the collecting duct, and to look for changes in that. In the course of measuring the electrical resistance, Sandy discovered that there was a big electrical leak out between the tubules and the pipettes that were holding them. And that problem was solved by Jerry Vurek, whom I've already mentioned, in Bob Bowman's lab, and his solution is shown on the next slide. The solution was to use a resin, viscous liquid resin called Silgard. And the Silgard coated both the glass and the tubule surface, making an electrically tight seal. And eventually what had started as a very simple arrangement shown in A here for perfusing the tubules became more complicated with the sill guard introduced around the tubule in B. And finally, the final arrangement is one in which rather than 2 pipettes concentrically, there are 4 pipettes concentrically, an outer pipette which has the sill guard, then one to hold the tubule, then one which is the perfusion pipette, and then inside of that a pipette for changing the fluid so that you could change the perfusion fluid going through the lumen in the middle of an experiment. And the other end of the arrangement, the collecting, also became more complicated, shown on the next slide. Again, starting with a Jared Grantham simple arrangement in A, then having a sill guard laboriously placed around the tubule, which is a little bit tricky in B, and finally another concentric pipette to hold the sill guard around the whole thing. This was a much more practical arrangement. These were more practical arrangements than they appear, because you could set them all up, and then if you cleaned things out carefully, leave them set up from day to day and week to week, which made the perfusion quite practical.
So there were hundreds or even thousands of papers published with this isolated perfused tubule technique that you invented. What do you regard as your most important discoveries using the perfused tubule technique?
Well, a few come to mind. There were a lot, but a few come to mind. One was looking at the part of the tubule called the proximal tubule, and discovering that the transport characteristics of that part of the tubule differed depending on which portion of it you looked at. For example, the early portions transported glucose very well and very rapidly. The late portions transported para-aminohippuric acid very rapidly and very easily. From that, a concept arose which I never held to. That was the concept of heterogeneity, that you could have heterogeneous parts within the same part of the tubule. And that was quite striking to me once when I spoke in Japan, and there was a banner over our meeting, which was mostly in Japanese that I couldn't read, but in the middle of the Japanese was the word heterogeneity. It turned out that there was no word in Japanese for heterogeneity, since they're very homogeneous, very homogeneous people. I found the term objectionable, as did you, because what it meant was that the refinements in transport along the tubule were simply greater than the anatomists had recognized, and that it gave additional control. And in fact, I think we wrote a review together in which we tried to trash heterogeneity as a term. I think we may have succeeded. I haven't heard it again recently. Have you?
No, not recently.
Okay. Then Bruce Toon, when he came, started on a ambitious program. Bruce, after his fellowship, went to Stanford, where he became the chief of pediatric nephrology. But his job was to remove the tubules after they were perfused and measure the concentration of the transported solute within the tubule cells. And he was able to discover the transport steps that way through the cell for glucose in the one case and para-aminohippuric acid in the other case. That, I think, later went on. Recovering the tubule became fashionable, and you had some very nice studies with Soren Nielsen, which recovered tubules, I recall. Then, in proximal tubules, the reabsorption of salt turns out to be very dependent on the reabsorption of organic solutes, like the glucose and amino acids that are present. And that was something I found when tried to get different perfusion solutions and bath solutions for proximal tubules. The problem being that we knew the rate of fluid absorption in rats from the micropuncture studies of proximal tubules, and was much less in our isolated tubules, which raised the question, maybe they're just not very healthy. I think there were two answers to that. One answer was that rabbit tubules don't absorb fluid as rapidly. Second was that in our initial perfusion solutions, we didn't have all of those nice organic solutes in the lumen that are necessary to get the maximal rate of absorption. And it's very striking if you do the experiment, if you start perfusing the tubule with a solution that has no glucose or amino acids in the perfusion solution, the tubule cells are very flat. The minute you put in the glucose and the amino acids, the tubule cells balloon out and become very broad because of the transport step when they're accumulated within the cells by the transporters in the lumen. Another controversial issue which we were able to address was the electrical potential difference across proximal tubules. That had been measured quite well in amphibian proximal tubules and found to be about 20 millivolts lumen negative. And the same thing was initially found in the rat proximal tubules. However, that was the wrong answer. And the problem was an artifact in which the pieces of tissue got stuck in the electrodes that were used to pierce the tubules. And of course, we didn't have that problem since we had an open cannula going into the end of the tubules. And so there's some interest in our results for that measurement. And what we found was that in the early parts of the tubule, dependent on the organic solutes, it was just a couple of millivolts negative. And the late parts of the proximal tubule, it was either negative or positive, depending on the contents of the tubule fluid. Shortly after that, Eberhard Frumter in Frankfurt did the studies correctly in the rat proximal tubule and confirmed our results in the isolated perfused tubule. Another issue, which is historical only, is that at that time there was a fashion for measuring fluid absorption from tubules by putting oil in them by micropuncture and then splitting the oil drop with a fluid that was to be transported and seeing how fast the oil drops came back together. And from that came the concept that the rate of the absorption of fluid was strictly a geometric question, and that the rate was proportional to the square of the radius of the tubule. I tested that in the isolated perfused tubules, varying the tubule diameter by varying the outflow pressure from the tubule, and found that there was absolutely no relation to the diameter of the tubule. It was quite controversial at the time, but I haven't heard what was then called R squared referred to in, I think, the last 30 years.
30 years,
30
yes,
years,
definitely.
The issue was settled at that point. Perhaps the most striking observation that we had was experiments measuring the voltage across thick ascending limbs of Henle's loop. And I measured it in experiments with Nordica Green, who was my technician at the time, and I know Juha Koko, who had been a fellow, measured it in Dallas, where he was perfusing tubules. And we both had the same reaction because we had all expected that the voltage would be lumen negative in all parts of the tubule, in the parts of the kidney tubules, and we found a positive voltage. And we went through the same thing, checking the leads, checking the polarity of everything. And finally it turned out that that was actually the voltage, and that led to the discovery of the sodium potassium 2-chloride transporter in the lumen of the thick ascending limb, which Nordica and I were able to discover was the transporter that was inhibited by important diuretics like furosemide. This is a picture of Nordica, who worked with me for many years. When I first came to the laboratory, she was working with Ernest Kutlov, who was studying chloride content of muscle cells. But when he left the laboratory, she became my technician, and she was a marvelous person, a marvelous scientist, and we accomplished a great deal together. It was a sad day for me when she decided to retire, and I could no longer work along with her. Another discovery was made by Dwight McKinney, who was a fellow and later went on to become the director of a big pharmaceutical company. Dwight was studying bicarbonate transport in collecting ducts. And everyone knew that bicarbonate was simply absorbed by kidney tubules. And what Dwight discovered was that whether it was absorbed or secreted by the collecting ducts depended on whether the rabbits from which the collecting ducts were taken were given alkali solutions or acid solutions. And this is now taken for granted, and there are many studies of the particular cells which absorb and secrete bicarbonate. But at the time, it was quite controversial. And I remember the experts in the field for many years greeted our discovery with, as far as I could tell, disdain, because they felt that it simply wasn't possible that this was going on. And finally, some early studies of Jared Grantham having to do with the route of water absorption through collecting ducts. And he found when he applied vasopressin to the collecting ducts that with a dilute solution in the lumen, that the tubule cells swelled up, which was the indication of the location of the channels, which eventually turned out to be water channels in the lumen membrane that are affected by the antidiuretic hormone. So there were many other things, but those are the ones that come immediately to mind.
So back in the early 80s, you decided to set aside the perfused tubule technique and go to other things. Why did you decide to stop?
There were two things. One thing was that you had agreed to stay on in the laboratory at that point, and you were doing a much better job of it than I was, so I might as well let you do it. The second was that science had moved on, and though I was obviously very fond of perfusing tubules, there were all sorts of new developments, and it was enticing to me to get my feet wet, so to speak, in some of these new areas.
So what areas in particular did you decide to pursue?
Well, decided to study osmotic regulation, which eventually comes from the fact that in the kidney medulla of animals, there are very high concentrations of salt and urea that may be poisonous. It's the question of what harm does it do to the cells, and how do they adjust to it?
Mm-hmm. And so what prompted you to go in that direction?
You're to blame.
Oh.
As you probably recall, at that time, Bob Balaban was also in the laboratory. Bob is now the director of intramural research at the Heart, Lung, and Blood Institute. At that time, he was doing, and still is doing, nuclear magnetic resonance spectrometry to analyze the intracellular compounds and their status, particularly things like ATP. You and he had a project together in which you decided that you would use NMR imaging to image urea in the kidney, and that you would do that using nitrogen, which
Nitrogen 14.
Nitrogen 14 imaging. The result was that for technical reasons that didn't turn out to be possible, but while you were doing the studies, you saw enormous concentrations of a nitrogenous substance other than urea in the kidney medulla, And Bob had been previously puzzling over enormous concentrations of a phosphorus-containing compound in the inner medulla. And putting that together, you realize that the compound was glycerophosphocholene, which is one of the principal organic osmolytes in kidney medullas. I think we all realized a little bit later that you hadn't discovered that had been in fact discovered by Karl Ulrich in the 1950s. Karl being a really talented German renal physiologist, marvelous person, good friend. Sorry to have him retired from research at this point and no longer being able to interact with him. But Karl found this compound in the kidney medulla. In a second paper, he figured out what it was, and he knew what it was doing there. And that was something none of us either knew to begin with or remembered. And so it was sort of a fresh discovery when you found it. So that showed that there was glycerophosphocholene in the kidney medulla. In 1982, Paul Yancey and George Sumrow, who were marine biologists, had published a landmark review on the organic osmolytes and how they're accumulated in cells in response to high salt and high urea, in their case principally in marine species, but they also in the review covered plants, and it's really a universal response. And in many of those examples, there's more than one organic osmolyte that's accumulated, and so decided to see what the other organic osmolytes might be in kidney medullas. And for that purpose, I was lucky to have as a fellow at the time Serena Bagnasco, who was Italian, had walked into my laboratory one day and asked if she could volunteer to work in the laboratory, her husband having a different job at NIH. Sure, I took her as a free volunteer, and it wasn't many months along the way till I realized that she's a superb scientist, and found a salary for her to stay on as a postdoctoral fellow in the lab. Anyhow, it was she had done other projects, but at that point it became her project to find out what the other organic osmolytes were. So she harvested kidneys from rabbits and rats that were either given a lot of water to make them diuretic and lower the osmolality in the inner medulla, or were deprived of water to raise the osmolality. Then came the question of how to detect the compounds. We're lucky there that Bob Balaban was willing to give us his NMR spectrometry expertise using proton NMR, and that we had in the Institute one of the world's leading analytical chemists who could do the same thing by mass spectrometry. And between them, able to define the other organic osmolytes, which were sorbitol, myoinositol, and glycine betaine. And so we published that all together.
Okay, and then many of your studies have depended on having cell culture models of renal cells. How did you get started with that?
Well, I'm not the one that started that. The one who started that was Joe Handler. Joe was in the laboratory almost from the beginning of the time I was, and for many years he was Jack Orloff's other fellow and collaborator who worked with him, worked with Jack. And Joe's specialty was toad bladders, in which he and Jack made seminal discoveries regarding the role of cyclic AMP in signaling vasopressin in the collecting duct and in the toad bladder. Well, after studying the toad bladder, we read a very interesting paper which by an investigator named Dayton Misfelt in California. And what Dayton had done was to take an existing line of tissue culture line from kidney, which was called MDCK, and to put it onto a porous support and to show that there was an electric voltage that was generated across it, giving the possibility of studying transport in tissue culture. Joe latched onto that technique. He trained himself in tissue culture. He had some of the early tissue culture models of transport, and he established tissue culture in our laboratory so that when I wanted to use tissue culture to study the organic osmolytes, it was already there. And I had just to learn the technology from Joe. We didn't have at that time any cell lines which were specifically from intermedullary segments. Nordica Green tried very hard with me to start some cell cultures from intermedullary and other parts of the kidney, starting with single tubules, and we were not successful. The reason was that that was before it was realized that there are certain genes which have to be expressed to immortalize cells in tissue culture, and we simply didn't have that information. But by luck and by hard work, Nordica had succeeded in starting a culture from the cells lining the intermedulla that are exposed to the same high salt and high urea, and we were able to use those cells in our initial studies.
So getting back then to the organic osmolytes, how did you use the cell cultures to understand the regulation of organic osmolytes?
Well, we looked in the cell cultures to see if they would accumulate the same organic osmolytes as in the kidney, and it turned out that they did. And this was marvelous for us, because that meant that although it would be very difficult to study the details of their accumulation in vivo, in the cell culture there was a direct route to doing it. And the initial measurements of the osmolytes in the cell culture were done with Bob Balaban in his nuclear magnetic resonance spectrometry. But about the same time, Paul Yancey came to the laboratory for sabbatical. I mentioned Paul once already, Paul being the marine biologist who was the expert in this area and who had co-authored the landmark review in it. Well, Paul worked with Bob Balaban to develop a HPLC method for measuring the organic osmolytes. That was quite practical and became the standard after that, so we were able to use that to study their accumulation in the kidney cells. And that's still the principal method for measuring them. And so we use those cultures to figure out the mechanisms by which the organic osmolytes are accumulated. And in those studies, had some more very talented postdocs. Two of them were Japanese postdocs. By that time, the Japanese scientists were coming to our laboratory to learn kidney physiology. They were Takeshi Nakanishi and Toshiki Moriyama. And they were able to discover the increased abundance of aldose reductase that was responsible for the accumulation of sorbitol. And Serena Banyasco and Shunyu Uchida discovered the aldose reductase. Takeshi and Toshiki were responsible for discovering that betaine and myoinositol are accumulated in the cells by transport, and that there were more transporters when you raise the salt concentration. Then Arlene Garcia Perez came as a fellow to the laboratory, and Arlene, who is originally from Cuba and Puerto Rico, had studied at MIT and Michigan in the United States, and had picked up some experience in molecular biology along the way. And she applied that in studies with the cell culture in her laboratory to clone the cDNA for aldose reductase, which was responsible for the accumulation of sorbitol, and to eventually show that that was regulated on the basis of transcription, which turned out to be the case for all of the other organic osmolytes. Then Mu Quan came along to the laboratory. Mu was actually Joe Handler's fellow, and Joe and Mu worked along with me and my fellows in looking into the organic osmolytes. And with Mu and Joe, we were able to use expression systems in toad oocytes to show that to clone the cDNAs for the myoinositol transporter and the betaine transporter. Mu, when he left the laboratory, went along with Joe Handler to Johns Hopkins, where Joe had become the chief of nephrology. They continued to study the organic osmolytes there, and together they cloned the transcription factor that's responsible for the accumulation, which they named TONI-BP. Joe has now returned from Johns Hopkins and is back in our laboratory and is very, very, very welcome here. Mu moved out of Johns Hopkins, is now a professor at the University of Maryland, and we continue to follow each other's work very closely since it's remained very much in parallel. Since then, we've continued to investigate the signaling pathways to TONI-BP, and those studies have been mostly conducted by Joan Ferraris, who had been a fellow in the laboratory for many years and is permanently with us, and is our molecular biologist and is responsible for uncovering those signaling pathways, which I'll not describe in detail since it would require too much time at this point. But it's very exciting to be following that through, and we have the advantage of the example in the laboratory, which is you studying systems biology, because it turns out that that's what we're getting into now, studying the systems biology of signaling of osmotic stress to the stress response proteins.
So tell us a little more about the damage that urea or sodium chloride at high concentrations cause in cells.
Well, clearly the cells in the kidneys manage because they survive and they function. But we wanted to look at that in cell culture, and we had striking results almost immediately. The background we had for that was that Yancey, again, and Somro had looked very extensively into the subject in marine organisms. And what they had found was that elevating the salt, which is what occurs in cells if you initially when you elevate the salt concentration outside of them, or elevating the urea, which goes into the cells immediately, has very large effects on the enzymatic activity of proteins and on the structure and function of macromolecules, including both proteins and DNA, and that the organic osmolytes help to protect against those effects. So we started looking at it in cell culture, and the one who carried out the initial studies, which were most revealing, was Dietmar Kultz. Dietmar started as a marine biologist, had his postdoctoral fellowship, first postdoctoral fellowship with George Somro in California. And when he came to our lab, he started studying the cell cultures from mammalian kidneys. And early on, he found that elevating the salt concentration caused an increase in a particular protein called GAD45, growth arrest and DNA damage inducible protein 45, which is our first indication of the sort of stress that occurs in the cells with high salt. Those studies were continued by more very talented investigators. Luis Mexia, who came as a postdoc from Chile, where he's returned as a professor at a university in Santiago. Natasha Dmitrieva, who came as a postdoc from Russia, from St. Petersburg, very talented scientist whom I'm fortunate enough has agreed to stay on in the laboratory and is one of my most valuable and valued collaborators and associates. And in this paper, in this same picture, Jane Zhang, who was a postdoc from China. And Lewis started studies looking at the fate of cells in culture when the salt and urea are elevated. He was able to demonstrate apoptosis, which is cell death, a particular form of cell death, and was able to demonstrate delay in the cell cycle, the rate at which the cells were growing. Natasha and Dietmar went on to show that one of the DNA damage responses, which is the activation of protein called P53, which is a tumor suppressor protein. And they showed that elevating the salt greatly increased the abundance and activity of that, another indication of the kind of damage that was going on. Jane, together with Natasha, showed that the cells suffered oxidative damage to their proteins when the salt or the urea concentrations were elevated. And putting that all together, Dietmar, after he left the laboratory and went to the University of Florida, where he was initially, he's now at University of California in Davis, where he's an associate professor. Dietmar was able to show directly that raising the salt concentration causes the DNA in the cells to be broken up, something which is incredible. And even more incredibly, Natasha later showed that the DNA in the cells in the kidney medulla, while the salt concentration is high, is also broken up. And those discoveries are the basis for studies that we have going on now, which are aimed at knowing exactly why these things occur and knowing exactly how the cells are able to protect themselves from that.
So in general, looking back, all of the people that you've trained, they've all done extremely well in their careers after they've left. What factors do you feel have allowed you to be so successful as a mentor?
You've exaggerated the case. The people who have done well were very good to begin with, and they went out and did well. I've had average fellows who've done okay, and I've had other fellows who were not terribly capable, and a lot of them did not do very well. So I can only take a very minor part of the credit. I don't think there's any particular system of mentoring which is more successful than any other. What I do think is important is the examples that are set for the fellows by the way things are run in the laboratory. I think the fellows learn by example rather than by being told specifically what they should do. In managing the laboratory after I became laboratory chief, I wanted very much to have a laboratory that was comfortable, as opposed, for example, to a laboratory where things are highly competitive and highly personal. I think we've succeeded in having a happy and comfortable laboratory where the stresses are scientific rather than personal. I think it's very important that the fellows are appreciated and get credit for all of their good and for their hard work, try very hard to do that. I think they have to be exposed to a laboratory atmosphere with the ethics of science are very much emphasized. They have to learn by example the importance of assigning the right authorship, the importance of being responsible when you review papers and when you review grants. These are all things which might seem to be very obvious, but I'm not sure they're the case in all laboratories. I'm very pleased that you've chosen to continue the same way now that you're chief in the laboratory. When I say a happy laboratory, I don't think that we lack scientific criticism. I think that we're very keen to the science, and we're very self-critical and critical of other people, but the criticism is always on the basis of the science and not on a personal basis of the people involved. And finally, I think by example, you have to show them that your standards of science are very high, that you want to have experiments that are done correctly, which are done reproducibly, and conclusions that will bear up in the present and in the future. And I hope to convey that to the fellows, and I hope that some of them have learned it.
I think they've all learned that. So in retrospect, then, looking back at all of these 40-some years in science, how do you feel about your career in science?
47 years.
47.
And counting.
And counting.
Well, you and many of the people I've talked to through the years have heard me gripe about working for the government and gripe about NIH. Forget that. NIH intramural program is a marvelous place to do science. And if the science is what really interests you, this is the place to be. You don't have to, you have a budget, you don't have to worry about grants, you don't have to spend all of your time doing that. You spend your time on the science. There's a certain responsibility there. The government has a big investment in you. I guess the government has invested somewhere between $50 and $100 million in my science in these 47 years. And I think that our responsibility at NIH, being given this money and not being required to apply for grants, is to try to do things that are truly innovative and that might be more difficult to do elsewhere. And I think that the tubular perfusion projects, and I think the organic osmolyte projects fit that picture, just as many of the things that you're doing now. And so what I feel is a gratitude to NIH and a satisfaction that I've chosen to stay on here. The science has always been very exciting to me. I like the science itself. I like the people I've been involved with. You have many brilliant people that you interact with at meetings and scientific organizations. It's marvelous to have enthusiastic, bright young fellows to have innovate in your lab and have be able to interact with them all of the time. And so my enthusiasm for the science and for NIH remains undiminished. And even though I'm well past the age where I could retire comfortably, I have no intention of doing it as long as my health holds up, as long as the science remains as interesting as it has been.
Well, Mo, thank you very much for this marvelous story and continuing story, as you point out. This is extraordinarily important that we record history as it occurs, and I think that it's been my great pleasure to be involved in this recording archival history project. Thank you very much.
And thank you for helping me to do it.